did it cost for them to get one of these, make one of these?
Yeah, I mean,
so the second one is to do with propolis on the microphone.
Yeah, the most expensive part at the moment is the scale, which is developed by Volkswagen, which is like costing €1,000, and you can also build this yourself.
Can.
And the rest is based on Raspberry Pi parts, so Raspberry Pis and cameras, Raspberry Pi cameras, and the microphone is the AudioMuff. I mean, at the moment, I don't know if it's really needed to capture ultrasonic sound. So you could also simply use a USB microphone, which is very cheap.
And with honeybees, they tend to smother the microphone in propolis. I've known some groups that have tried to find a solution to that. Is that a problem with bumblebees? And if so, how have you dealt with it?
Yes, I placed like a plastic bag around it, but
that doesn't attenuate the signal too much.
No, I don't think so. But I mean, as the colony grows, so the colonies I placed really in the field, I let them grow until the end, and the boxes were full, and so they also started to eat this plastic. And I mean, yes, they were really dirty.
Thank you.
Also, two questions. First is, just I saw that you mentioned about you are using the common pesticide. So do you know what is which group of the pesticide that you're actually using?
Yes. It's one herbicide, one fungicide, and one insecticide. And
no, I mean the functional group, like for example, neonicotinoid.
Yes, neonicotinoid. Yes.
So that would be the key point that would affect them. So that's why which group that you are using?
Azoxystrobin, terbutylazine, and acetamiprid. Those three groups.
So are those like they are the below tuning concentration.
Yes, they all didn't die, but I mean, it was also a big problem because there are not so many studies on bumblebees about what concentrations are lethal and which are sublethal and at what level. So we had to guess, and I mean, it would have been nice to have a pre-study to decide on which concentrations to take.
And the second question is about bioplastic. I saw the spectrogram that you have is like whole range. So then, what your specific frequency that actually can capture that wind beats?
Yeah, like the bee sound is quite low. It's usually at around 200 hertz or something. But I mean,
it comes from the thorax vibration of the wings.
But we see these pitches that go really high up until the ultrasonic range. So it's very, I mean, I cannot listen to this sound because I'm a human, but I see these on the spectrograms, and I wonder if they are, of course, important for detecting stress. Yeah.
Some because you can spectrograms can be can change with spectrogram and changing the time.
Okay.
Try to some kind of representation is less exactly.
Okay, you mean like do some filtering or something on the data?
And also the time signals tends to be fairly stable over a short time scale, so you can adjust the window for that way. Doesn't tend to change over the course of seconds.
Yeah, but I think it's hard to just remove them, because if I remove constant lines from my recordings, I would remove the bumblebee sound as well, and that I don't want. So
So thank you so much. If anyone has any more questions, please go over them during the lunch break. And now let's continue with our next presentation from Gordon Hunter.
So good morning, everyone. So this is work primarily done by my recently completed PhD student, John Butcher, who is firstly a beekeeper and also older than me. So he's retired now, but so he just fancy doing this out of interest. And I'm at Kingston University on the edge of London. So as you may know, and from that previous talk we got well primed, insect pollinators are vital for both agriculture, because most fruit crops, including things like tomatoes that are really fruit, and just about everything except cereals really are pollinated largely by insects. So they're vital for our food supply. general biodiversity as well, because a lot of flowering plants are pollinated by insects as well, and they contribute to various other things, including medicines and biofuels and construction materials as well. So many trees will be pollinated, not all, but many trees will be pollinated by insects. So there has been a severe decline, as you probably know, in insect populations due to multiple factors, possibly climate change and environmental pollution, certainly due to pesticides and so on, but also due to parasites and predators, including a lot of invasive species. Now, major parasites of bees are mites. They're strictly speaking not insects themselves, they're arachnids. And these can spread bee diseases, in particular the Varroa mite that we see here. And this is very much magnified because it's about 1.8 millimeters that way and 1.2 to 1.4 that way. So this is large magnification. You see a few here. Is this a pointer? You hear individual spots there on bee pupae. These are individual Varroa mites. They're parasitic on the pupae, on adult bees as well, but particularly on the pupae, larvae and pupae. But they also spread a lot of bee diseases, in particular deformed wing virus. It does exactly what it says, deforms the wings so that they can't then fly. So these are considered a real major threat to bumblebees as well, but particularly to honeybees. And so they need to be monitored. They're endemic now on every continent except Antarctica, where there are no bees. They weren't present on Australia until quite recently, but they are now in Australia as well. So there's several ways you can monitor for these, but some of them require killing some of the bees. So there's various things like alcohol washes and sugar shakes and that are generally quite accurate, but estimates of the Varroa infestation levels. But they require sacrifice to some of the bees. This method, which is quite commonly used, I think we found that in a fairly big survey of UK beekeepers a couple of years ago, where we got over 1,000 individual responses from a wide range of areas across Britain, including Northern Ireland and Scotland. We found that about 80% of the respondents used this Varroa board as an approach to monitoring their Varroa infestations. So this is a Varroa board. It's about 1 meter by about 60 centimeters, goes under the hive. This sort of wire mesh that stops individual dead bees falling onto it, but you will get various detritus, various rubbish falling through, including some dead bee parts, quite a bit of propolis and so on, which is the secretion that the bees use to seal the hive and so on, and dead Varroa mites. So you've got a mixture of all sorts of stuff on this board. So John's idea was, well, if you try and take these out and inspect them near the hive, if it's a windy day, then they might blow all the dust and stuff, including the Varroa, away. Can you just take some mobile phone images quickly that you could then analyse? Because you can, if you try and count these individually with magnifying glasses and tweezers, it will take a long time and it's quite hard and demanding on your eyes and so on. So he did this and he found initially an online tool. I'm not sure if this is the best one or the worst one on the market, free online tool. He took this photo of one of his Varroa boards. He fed the photo into one of these online tools and it told him there were 456 Varroa mites. It wasn't a windy day, so he was able to count them and he found 15. So it's a factor of 30 too high. It's obviously not satisfactory. There may be better ones out there. This is just one example, but can we do better than that? So to start with, he tried using classical approaches to machine learning. So this is a Varroa board, and that little circle there is a metal washer, which we see magnified here. So this is a close-up of part of the Varroa board, the same board. And there's one Varroa mite there. There's some other bits and pieces here. There's probably a Varroa there, possibly one there, but that one's almost certainly a Varroa, that one less so. So you get bits of stuff, propolis and things like that might be mistaken for a Varroa. So we did some classical machine learning approaches on this. We had originally 450 items, only 32 of them were actually Varroa initially. So we used various things like shape factors, color factors, and size of the object as manually to derive features. So this is some of the image pre-processing here. First of all, silhouetting the image and then approximating it by an ellipse, and then looking at the difference between the silhouette and the ellipse and so on. We got quite good results with, well, pretty good results actually, with classical machine learning. The best one was a form of regression tree approach. Here, this one called Mite, multiple additive regression trees. We got high accuracy, good recall, and pretty good precision on test data. We got very few false negatives, but quite a lot of false positives. But we needed to have close-up images for these to get good resolution, taken for about 10 centimeters away. Here's an example of a false positive. This blob here, roughly the right color, roughly the right shape, but not perhaps a bit too rough compared with a real Varroa mite, and roughly the right size. It wasn't surprising, it was a chunk of propolis. It wasn't surprising that got mistaken for a Varroa mite. Not quite the right shape, but not too far off. So these were very close up, very high resolution images. There you'd have to take multiple images of the board, and on a windy day that might not be appropriate. But so can you do better with either half board or whole board images? So we tried using various deep learning approaches, some CNN models. Oh, sorry, I've gone the wrong way. Some CNN models and a visual transformer model as well. This was, which one was this? I think that was, yeah, that was the visual transformer that was the best performing of the lot. We were getting very close to perfect. This column here is the visual transformer. This one is the best performing CNN, which was ResNet 50. We tried various other ones, including Inception and VGG16, I think it was. We tried a few different CNNs. This was the best of the CNNs, performing pretty well, not quite as well as the visual transformer. So again, we were getting far fewer false positives this time. And this was with, some of them were half board, some of them were whole board images. So you only had to take one photo, or possibly two per board. So what have we found so far? Well, as you probably know, honeybees are very important in ecology and agriculture. They're threatened by diseases spread by parasitic mites, including Varroa, which is widespread across most of the world now, particularly in Europe. And you need to control for these, otherwise the colony is likely to die out within about 3 years. Some people claim they've got Varroa-resistant bees, but they tend to be in quite isolated regions where maybe they're just not getting enough contact with other bees to spread Varroa very extensively. So we're hoping to try and extend this work, more data, and sorry, spelling mistake there, apologies for that. And also a new threat, another mite spreading right now. So the top one here is a Varroa mite. So that's the same, as I said before, about 1.8 by 1.2 millimeters. This lower one is this new, I always have trouble pronouncing, I think it's Tropilelaps, however you pronounce that word. I check my spelling of it, but I always have trouble pronouncing it. So this mite is spreading into Europe now. It's spreading west. It's reached Georgia and the coast of the Black Sea, parts of Russia bordering the Black Sea, possibly spreading now into Ukraine and Turkey. So it's coming towards Europe. And that's got a faster breeding cycle. It's believed to spread similar set of diseases to the Varroa type. And so we're trying to think about, could we help to do that? And we've done some work with other things. So just some acknowledgements, and thank you very much.
Questions? Yes.
I just have an idea for you.
Mhm.
So A lot of arachnids, ticks included. I think these mites as well are fluorescent. So if you took a photo, like it's dark, and you put a UV light, like a 365, 395 nanometer light, they just glow. It'd be like bright spots, and everything else would be dark.
So you use the light in when you're taking the photos?
Yeah. So take a photo in the dark.
Yeah.
You need to be on a tripod probably because it's going to be longer exposure. But the mites will be like bright neon colors, and everything else will just be black.
I didn't know that, so that's a useful bit of information.
Yeah.
Thank you.
With the NBU in the UK?
Not directly, but I actually know someone that is employed by them or by DEFRA, the Department of Environment, Food, and I can't remember what it's the Rural Affairs. Thank you. And as a bee inspector, so we are liaising with him. But he's particularly in summer, he's extremely busy because he has to cover a huge part of England going to do inspections. So he hasn't had that much time to devote to working with us. But yeah, the idea is to work. But we are working with a lot of, well, certainly local beekeeping associations in our area. And the survey was sent to, I think it was 250 roughly local beekeepers associations in the UK. And we got, as I said, we've got a very good response rate of over 1,000 responses. So, you know, there is opportunity to disseminate this work through the National Bee Unit and so on.
I don't know much about this question. They also spread to solitary bees, which are quite important for you.
Yeah, some of them aren't affected by Varroa. Certain bumblebees are. A lot of them do have parasitic mites. Parasitic mites are very widespread across different types of bees. And I don't know how much work has been done on it, but I'd imagine they could spread disease as well that might affect those bees. I know someone in Slovenia that works a lot on solitary bees. We're trying to do a collaboration with them. But thank you.
That's all. Thank you, Gordon.
Thank you.
Our next presentation is from Iris Puchlovec.
Possibility to talk here, and I would like to say something more about all review. I was approximately before 1 year asked or request that I wrote something about ladybirds and artificial intelligence. So I invite few friends, now they are co-authors, and they help me with to make such review. The final we a little bit change it, and it's the role of computer vision in monitoring ladybirds. And I like stories, and I think that everything is necessary to start with some broader part and go to some aim, but the aim is not necessarily the final point. It's necessary to look also to behind, in fact, look for future. So that's what I speak about. So firstly, before a few minutes, you heard many things around this, but firstly, why care about insect? From environmental side, we see, and it's completely confirmed that it's here some insect decline. It's completely not only on biodiversity, but it's also about abundance of really general species. So this is very important, but not each people are interested about some environmental, but many people are interested about economic side. So it's also to show them that it's here is some economic part. Yeah, it's that insect takes some role in ecosystem roles like pollination, predation of pests, predation of weeds, so decomposition. And here is direct link not only to agriculture, but also to forestry, to fisheries, for every role. And this is also ecosystem services have very good connection with food production. So now that some people are start to listen. And my favorite group are, of course, ladybirds, because they are cool. So, and why we work with ladybirds specifically? It's a relatively big group. It's 6,000 to 7,000 species. There is the view of the taxonomist, because they including some subfamilies or not, or excluded. Very good for ladybirds. They are relatively sensitive, not only to climate, but also to land use, also to pesticides. So one part that we needed to inform how it's what is happening. So that ladybirds are using for model organism, and what is most important, they are really culturally popular. So we can use citizen science because ladybirds are cool. Everyone knows. Every culture has some specific knowledge about not that they are health, that they are predators, but every culture. For example, in UK, you have special company for all books for children are labeled by ladybirds. In Czech Republic, children's singing to ladybird, they are going to the god, taking the notice. So every culture has something for ladybirds. So they are very important. So it's very good to, it can be like indicator species concept for this part, but it's excellent bridge between science and public. Yeah. So why monitor ladybirds? In several countries, there are really long tradition to monitoring ladybirds, like UK or Czech Republic. So we have clearly detected that there are some declines of some species. Then it's also a very good example of invasive species, because ladybirds are normally very used for biological control, but there is a really nice example of negative effects of biological control, and it's Harmonia series. Use it against some pest, but there was something happened, and now it's problematic species. So what can we do? We still have some limits for traditional monitoring, and it's necessary to make some new steps how to work it. So what very maybe nonsense question here, what is computer vision? So I little bit jump across the technical sides, and I see something more from ecological view or for a view for citizen science. So here are tasks like we need detect species, identify, not every time, but also what is very important for ladybirds, to count individuals. Because ladybirds are very often used like biological control against also in greenhouses. And you can make some models. Then you put some ladybirds inside and they destroy almost all pests. So it's not only about computer vision, but they are next steps. And of course, we needed some labeled images. We need large data sets. And the vision is go from some manual training to some automated ecology. Yeah. So what is cool for ladybirds for computer vision is they have very strong visual patterns. They have a really nice colors. They have special spots, different version. So it can help us. And what is also good that we have already have many photos. They are available from many citizen science projects. What is also good that they are easy detection compared to cryptic species. Of course, there are also some exceptions like genus Skimnus. They are really small, black, and no one is able to identify it. But majority is really nice coloration. But existing also some problems like polymorphism, ladybird. That means this all is one species. It's a Harmonia series. And of course, it exists more forms. So it's also necessary to have more knowledge and more data sets for this specific species. So of course, there are some advantages for computer vision and some new challenges. I choose only a few of them. For example, bias. If we use computer vision, there is no observer bias. Yeah, that it's really going down, because if you have experience in something, you go directly to something and this is going out. But also it's challenge for bias, because rare species are of course underrepresented. Yeah, because it's not so easy to get the photos. But advantage for my view it will be a really nice potential for continental scale monitoring. That will be we have more and continuous working on it. Yeah. Of course, challenge it's also when we have some photos from the field, that the question about data quality we see on previous presentations, that is not so easy. For example, for mosquito, if you make some really nice lab photos, it's perfect. but when you got some photos from the field, it's not so easy. Yeah. Especially for rare species. Here is also a thing which is only for a short time, this slide, because everyone see it. If you was not so lazy like me yesterday or tired, so you can see many camera traps there and you see how what is possible. But of course, as I said, here are examples on in-situ cameras, but for ladybirds can be really nice to start with lab imaging system in greenhouses because they really need it. And there are some companies we really like to know not only for species detection or species identification, but counting of abundance. Yeah. So it's very important, some continuous monitoring, especially in the greenhouses, but there are also some trade-offs, some costs of scalability, how to work with it. So what is very important is the possibility to use citizen science and combine with artificial intelligence. Before several years, we made with some people who are in counters like Kelly or Ellen Roy and some others, we make the European Ladybirds app. Firstly, we thinking that we want to educate people. So we not use artificial intelligence, but we make some special key or filter key that people will be educated and how they can recognize the ladybirds. But later it was seen that people are not so interested about it. Still there exists some group who want to use it, but the majority of people want to some easy way, so they use it as more e-naturalized. So it's possible to use it both, but e-naturalized is very quick. AI helps with identification, of course, people validate. So now there are a million of records and it's possible to use it for some algorithms. What is strange of citizen science? Of course, it's massive data. That's perfect. And as I say, ladybirds are easy for them. What is weakness? It's rare versus charismatic species, because if something is really nice color, you make photos of this, everyone, so you have 1,000 photos, but this one, the black one, it's not so easy, or people don't like it. Also about location, because not everyone going. And now the future view or application ecology, I speak a lot about it, about the predict population dynamics. So it's a very important part, I think, to using in IPM, early detection of some invasion species, what is used for example, Harmonia axyridis. But what is also important for next steps to use some climatic data to models, and because we see everything is changing. That's on the stable part, that everything is still changing. And we must still apply the new things. And I think that AI can really help us in this. What is necessary to discuss? It's for future some digital twin concept. It's mean real-time ecosystem monitoring. And what is also important also to discuss with people. And we can support for farmers, and then will be help for agriculture. Our future perspective, it's integration, artificial intelligence, ecology, and many different sensors. It's a really cool now. Then we can get some real-time monitoring system, and then we can make some better models. Yeah, it's shift more to adaptive management, and what is really important, and I think that discussed action is really show this, is interdisciplinary work. You need much more complex of people, ecologists, data scientists, computer vision, also psychologists, because if you work with the farmers or with the people, not citizens, it's necessary to understand them and fill the gaps between us. And some take-home message, I think that's computer vision. It's a really cool tool, almost like ladybirds. And it works for ladybirds, and it enables like large-scale monitoring, faster detection, better ecological insight. But still, there are some challenges, especially data quality and bias. It remains still critical. And I think that it's still necessary to work with it and thinking about it. So my final line idea is computer vision will not replace ecologists. That's also very important to speak to ecologists. You will not replace, we still need you, but your role will be a little bit different. You will have more time for something, for making models and everything around it. That's also very important to discuss with people. So thank you for attention. That's my favorite figure to show that it's necessary to several view. Many people like cool ladybirds, but not aphids, for example. So thank you very much. And one last thing, I was requested to inform you that exists also some alien violets. It's already, this year will be the 5th year. Before 4 years we started, it was almost 13 countries who cooperate with us. Now it's 6 or 7. So if you want to, or your country, join us, ask me or Kelly, who are coordinators in Europe. So you are welcome. So thank you for attention.
Thank you, Yiri. Any questions? No, that's all. Oh, one.
No, there is no model. There is no model. We make only some.